Hi, everyone. I hope you're doing well. My name is Christopher Good, and I'm really excited to talk to you today about my honors capstone project, the title of which is Using Blasts to Detect Horizontal Gene Transfer in Pathogenic Fungi. And I completed this project under the direction of Dr. Teresa Grana in the biology department. In terms of rationale, my interest was definitely piqued in my bioinformatics course with Dr. Grana when we participated in a community science project developed by a team called Genome Solver, and they have used a Python code to develop this BLAST or basic local alignment search tool pipeline in order to be able to detect potential instances of horizontal gene transfer. And in this project, we were looking at horizontal gene transfer between bacteria and bacteriophages. Then later in the course, we read a research article about microbial dark matter. And then in the context of this article, it was about the failure to identify causative pathogens in cases of neonatal sepsis. Uh, this inflammatory immunosuppressive condition resulting from untreated infection. And the article really described how scientists are currently using metagenomic analytical tools to further understanding of of microorganisms that are pathogenic to humans. And that definitely piqued my interest because um, throughout my studies in biology, as a graduating senior now, we've talked about microbiomes a few times, and I've always found them to be really interesting because the composition of microorganisms that inhabit different parts of the body, they play important physiological roles and important roles in a number of processes like immunity. So as I did more background research on cases of sepsis and when the causative pathogen can be identified, it seems the majority are bacterial. Of course, there's also a fungal minority, and I think that's worth pursuing. So horizontal gene transfer is way more common to prokaryotic organisms, but in terms of eukaryotes, there is limited evidence of horizontal gene transfer in species of the Candida genus, a type of pathogenic yeast. And those instances were between Candida yeast and bacteria, and between Candida yeast species and another yeast species. So I think that warrants additional research on the evolutionary forces behind fungal pathogenesis. And quickly, Candida became the culprit of interest because they are an opportunistic pathogen. So they do inhabit certain microbiomes, but it seems, at least from when I was doing research, that when some kind of disruption occurs, like an invasive surgery, that can open the door to infection. So the essential questions in this project were what bioinformatics tools can be used to not only predict potential or other potential fungal pathogens, but also, more importantly, what is the role of horizontal gene transfer or fungal horizontal gene transfer in the context of antifungal resistance gene acquisition? And in terms of the methodology, I started by using Blast, and of course, Blast is a software. There are a number of platforms that support it, but I just prefer it just because the interface I think is easier to work with. The National Center for Biotechnology Information, or NCBI, their BLAST program. So I used previously sequenced antifungal resistance genes, and they're each, in bioinformatics project, sequence data, it has an accession number attached to it, and that's just like an ID tag, if you will. And the antifungal resistance genes that I searched in BLAST, in starting with a number of Candida species, were ERG11, ERG3, TAC1, GSC1, CG, FSK1, and FKS1 and FKS2. And in doing the blast search, so I had to start by doing a forward blast, starting with the fungal sequence and seeing the viral hits that appeared, as well as a reverse blast. So going from those viral hits back to the fungal hits and potentially getting the same hit. And then in a bioinformatics project, this is just one thing I've found, and I think this holds true, is there's a lot of data that comes at you, and it's almost like a fire hose. So it's really important to establish cutoffs and know what data is actually worth exploring further, or could indicate something. So in this project, the BLAST cutoffs that we used were greater than 40% query coverage, an expect value of less than 10 to the negative fifth, and originally we wanted to see greater than 80% identity. Of course, we were a little flexible on this because for eukaryotic horizontal gene transfer, comparing fungi and viruses, just because they're so different, we place less emphasis on percent identity in this case. So through the first month, it quickly became evident that the strongest BLAST evidence for fungal horizontal gene transfer, or the strongest antifungal resistance gene, was ERG3, which the gene product is C5 steryl desaturase, and that's going to play an important role in ergosterol synthesis, or cell membrane stability. And going forward with this, the next month we used BLAST, going from pathogenic fungi, looking at the PEP8 gene, because that's a confirmed gene in fungal pathogenesis, and looking at other fungi. And that did not come up with a lot, at least not any fungi that are known to be pathogenic to humans. But just to be thorough, I also did the same forward and reverse process in those fungal hits, and it's with a special interest in ERG3. And of course, that, in terms of the BLAST evidence, didn't reveal a whole lot. So a BLAST, because it's a quick statistical tool, other research articles often say, to have more support for horizontal gene transfer, you really need to analyze the data further. And in doing this, I used, first I constructed an alignment, because that's just a quick visual way to look at evolutionary conservation and sequence data. And I used that with using a program by the European Molecular Biological Laboratory, or EMBL, the program called MUSCLE, or Multiple Sequence Comparison by Log Expectation. And that is featured on my results next to the phylogenetic tree, which I'll talk about right now. And for that, I used MEGA, or molecular evolutionary genetic analysis. That's a pretty commonly used software. And in doing this, I constructed a maximum likelihood phylogeny of the antifungal resistance gene of interest. And in bold are the hits from BLAST evidence. And what this can do is tell us the likely evolutionary relationship of the sequence based on the alignment. So in terms of the BLAST evidence that was strongest, ERG3 in Candida albicans and Candida californica came up as a hit for the forward and reverse with Orpheovirus, and Candida glabrata came up as a hit for Adapus virus. And both of these viruses are actually a type of giant virus and known to infect algae. So that was kind of surprising. I kind of figured a mycophage or even a bacteriophage would come up as a viral hit in the BLAST searches. But in pursuing this, I also thought it was worth, and also for the purposes of the phylogenetic tree, because it's important to have an outgroup, I also included the algal host of these viruses, which is Acanthamoeba castellani, and that is at the very bottom of the tree. So in producing the tree, it quickly became evident that the maximum likelihood phylogeny results contradicted the BLAST results, in the sense that the ERG3 gene in the Candida species were pretty much grouped with yeast, not in a smaller in-group or closely related with the viral ERG3 orthologs. And what was kind of weird, though, was another fungal species that was a PEP8 ortholog, so of that pathogenic gene that encodes a sorting protein, that Hypopygia bertoni, this kind of unsuspected fungal species, ERG3 in that species was grouped closer with the virus, or with both viruses, than it was with either the pathogenic Candida species or with the algal host species of both viruses. So I tried to pursue that, and in BLAST, that didn't come up with anything. But a reason for that could be due to the length of the ERG3 sequence. And of course, this is the protein or the gene product, the protein sequence. So that's around 370 amino acids. So not a terribly long sequence. And that could explain those results. And then I will move myself over to the other corner and conclude this presentation. So in conclusion, while BLAST showed limited evidence of horizontal gene transfer of ERG3 between the three Candida species, which were Candida albicans, Californica, and glabrata, with the two viruses of interest, Orpheovirus and Adapus virus, the maximum likelihood phylogeny did not support these results. And of course, that's based on the alignment, and I did not include the full alignment on this poster. I only included the enzyme active site alignment for ERG3P, which is located inside of a histidine box, according to one article. And of course, the one species there, the one fungal species, there was no observed histidine box, but that is what the alignment came up with. But it didn't support the BLAST evidence, which is really strange. So considering that, it's likely that BLAST, for the context of horizontal gene transfer, is probably better suited for prokaryotes, not eukaryotes like this project. And it's important to remember that it's a quick statistical tool, so it's not really meant to be a definitive predictor of these evolutionary events. And another thing, or important thing to consider, is that because less than 30% of Candida albicans, so a really more common fungal pathogen, considering that less than 30% of its genes are characterized, and that value is going to be a lot smaller for other Candida species or less known fungi. Really being able to predict more gene products and characterize the full genome of other fungal species, that could reveal new evidence of horizontal gene transfer. So it's, while we didn't find a definitive instance of horizontal gene transfer, we found BLAST evidence for ERG3 horizontal gene transfer that seemed to contradict the maximum likelihood phylogeny prediction of ERG3 horizontal gene transfer. So I'd also like to thank the biology department and the honors program at the University of Mary Washington for giving me the opportunity to conduct this project. It was a really amazing opportunity. And I also want to thank Dr. Vinyak Mathur at Cabrini University. So he's part of the Genome Solver team, and his expert guidance was super helpful in completing this project and making sense of this data. And that is all I have, but thank you so much for listening, and let me know if you have any questions, I guess, in the comments. Take care.